I hope you can hear now. A very warm welcome to the 20th anniversary seminar of Finnish Theraplay Association. Eli tervetuloa Suomen Theraplay-yhdistyksen 20-vuotisjuhlaseminaariin.
Pistä lähemmäs. Okay, so we have an interesting day ahead of us. And in the beginning, we just wanted to give everyone, even those who are not so familiar with Theraplay, some idea what Theraplay is and the history of Theraplay in Finland.
Well, actually, we will hear more about the history later because we are very honored to say the founder of Theraplay Therapy is here today, present with us. So welcome, Phyllis Booth. It's an honor and a privilege to have you here again. I don't know how many times you've been here, but it's many. And we will go now over our program a little bit, what we have ahead of us for today. Eli tervetuloa toivotettiin Phyllisille ja nyt käydään vähän läpi tätä päiväohjelmaa tässä Hannan kanssa. Ja me ei esitellä Theraplayn historiaa varsinaisesti. Phyllis ja Sandra Lindaman tulee kertomaan siitä lounastauon jälkeen meille hieman tarkemmin. We will start with some very, I would say, hardcore scientific presentations by two world-leading researchers coming from Turku University. We are very happy to have Hasse Karlsson and Riikka Korja as our main lecturers today.
Eli toivotetaan tervetulleeksi Riikka Korja ja Hasse Karlsson Turun yliopistosta. Ja he kertoo meille aivan mahtavasta tutkimuksesta ja me ainakin Saaran kanssa odotetaan sitä todella paljon. Ihana kuulla siitä, mitä te olette tekemään.
I think it's something we all need to know. Many of us are clinicians here, right? Yeah. So we work with parents, we work with children in early interaction. I think we need to understand why. What are the principles? What are we trying to influence and what are the mechanisms? Ja siitä me kuullaan ihan hetken päästä. Eli hermostollisesta pohjasta. Miksi me työskennellään varhaisen vuorovaikutuksen kanssa? Mihin me yritetään vaikuttaa ja fokusoida? Sujuvasti käännetään itseämme. Meillä oli hyvä sopimus. Toinen puhuu englanniksi, toinen suomeksi. Tosi hyvin menee, Hanna.
Kunhan puhutaan.
Then we will move on to more details about Theraplay therapy. So we will hear about the history of Theraplay.
Ja sitten 12 jälkeen yhdeltä siis kuullaan sitten Phyllisiltä Theraplayn historiasta tarkemmin.
And then we will present some research findings about the effectiveness of Theraplay. What do we know from research point of view?
Ja sen jälkeen kuullaan vähän vaikuttavuustutkimuksista Theraplaysta, eli siitä, mitä sinne tutkimukseen tällä hetkellä kuuluu.
Ja sitten mennään puhumaan niin kuin laajanäkökulmasta. Jukka Mäkelä luennoi meille Theraplayn suhteesta kehitystieteisiin. Miten ymmärtää Theraplayn vaikuttavuutta?
And then 2:45, Jukka Mäkelä will tell us about Theraplay from the perspective of development science.
Okay. And at around 4 o'clock we will close with some words from our current chair, Lea Mattila.
Ja 4 sitten vielä loppusanat. Lea Mattila, meidän puheenjohtaja, kertoo meille sitten vielä jotain kaunista.
Many of you know, but all may not know, so just to anchor you into today. What is Theraplay? So Theraplay is an active attachment-based parent-child interaction intervention. It aims to promote structure, nurture, challenge and engagement within a parent-child relationship. You want to translate?
Mä luulen, että haluaako joku, että kyllä mä voin sanoa, mä ajattelin, että varmaan aika moni tietää jo tästä, mutta siis Theraplay on kiintymyssuhdepohjainen hoito, ja nimenomaan lapsen ja vanhemman väliseen vuorovaikutukseen tarkoitettu. Ja meidän 4 kulmakiveä ovat jäsennys, haaste, yhteys ja hoiva.
The unique aspect of Theraplay is the way we try to increase these dimensions, and that's true playing, that's true non-symbolic playing, making recorrective moments happen between a parent and a child.
Eli Theraplayssa on erityistä se, että me käytetään leikkiä ja ei-symbolista leikkiä nimenomaan, jotta me voitaisiin luoda vanhemman ja lapsen välille sellaisia korjaavia yhteyden ja niin kuin hauskanpidon kokemuksia.
We work very hard with the parents. This is really for the parents. We are doing all the playing and all the activeness. We want to create them a new sense of how to be with their own child.
Me työskennellään todella aktiivisesti siinä vanhemman kanssa, mutta me ollaan myös aktiivisia tekijöitä näyttämään, leikkimään, mahdollistamaan niitä hyviä hetkiä ja luomaan uudenlaista ajatusta myös vanhemmalle siitä omasta vanhemmuudesta.
And just to get you guys into the mood, we will show you just very short video clips and then we will have some nice morning activity to start. So this is therapist. Can you see it? Yeah.
Noin vahva, vahva, vahva. Niin. Pistäs kädet tälle pystyyn. Ja nyt kun mä sanon 1, 2, 3, niin työnnä nyt kunnolla. Ootko valmis? Okei. 1, 2, 3. Toden totta. Mahtava poika. Aivan mahtava. Mitenköhän ne jalat toimis? Onko jaloissa voimaa yhtä paljon? Ja odota merkkiä. Ja nyt kun mä sanon nyt, niin lähdet työntää. Ootko valmiina? Hyvä. Odota merkkiä. Ja se tulee nyt. Voi veljet, sä oot vahva. Voi veljet, kuinka vahva poika. Ja vasta 4-vuotias. Aivan mahtavaa.
Yeah. So this is essence of Theraplay, right? It's fun. It's creating a sense of mastery and competence. I can honestly say it worked. Thank you, Ilana Vierikko, one of our senior trainers. And the reason why I can say it worked is this is my son. When he was 4, he's now 12. He's a very competent and strong little boy. So it really worked. Thank you for this Theraplay session. Okay, and play, we want to emphasize, playing is important with young children, of course. Also for older children, who have depressive problems, anxiety, a lot of problems with their self-esteem. Playing with an adult who tries to increase their sense of strength and competence, it's equally as important. Okay, so let's see an example of a therapist playing with an older child, and we want to remind you where the parents would be. They would be observing this at first, observing, just
Nyt, stop. Nyt. Apua. Stop. Nyt. Yes. Good.
and with girls too, of course.
Stop. Jep, hyvä. Lähdetään liikkeelle nyt. Eikö se osu poskeen?
Ei osunut.
Ei saatu, okei, hyvä. No aina pitkät kädet, saa aikaa. Hyvä. Stop.
Okay.
Yes.
All right.
And parent was observing. That's why the camera is moving, because I'm laughing in the corner. I'm filming that. Those are my kids.
Right. So that would be really structure, that would be engagement, that would be challenge. But obviously, of course, what we then need to do, and we want to keep that in mind, we need to transfer these experiences really to the parents. That's at the heart of Theraplay, okay? It's not just for us, it's fun to do, but this is for the parents. So the mom is with the red shirt, of course. All right. So Jukka and Eira, thank you for you guys too. Yeah, we are ready to start. Just a couple of practical reminders still. We have a video recorder in the back who will be filming our day. We will make an edited version of the presentations and that will be available online. Sanoiko sä suomeksi?
Eli täällä on videointi käynnissä tänään ja tästä tehdään semmoinen editoitu versio, joka tulee sitten nettiin saataville.
Joo, ihan kaikkien saataville sitten jatkossa. Slides and handouts will be available later to you guys, all of you who have registered into the training, so you will get them after the training. We appreciate if you're quick with the lunches and the breaks, because we want to keep our schedule, so please come back in time.
Tämä on tärkeää sanoa suomeksikin. Yrittäkää olla oikein täsmällisiä. Meillä on ne 15 minuutin tauot erityisesti, mutta myös lounastauot. Meillä on paljon asiaa ja me toivotaan, että me saadaan se kaikki teille välittää. Niin se vaatii sen, että olette täsmällisesti takaisin täällä taukojen jälkeen.
Okay. There are books, there are brochures, information in the hallway. I think most of you saw that when you came, so take a look at there too. And we want to thank you all for coming. I'm Saara Salo and
I'm Hanna Lampi.
We are here as to welcome you from behalf of the Finnish Theraplay Association. So warmly welcome to the day.
Eli tervetuloa vielä kerran. Täällä on voitte käydä kurkkaamassa vielä tähän aiheeseen liittyvää kirjallisuutta tuolla aulassa. Siellä on semmoinen pöytä, josta voi katsoa ja jopa tilata. Ja kiitos kaikille, kun olette täällä. Lämpimästi tervetuloa. Hyvä.
Mä laitoin tänne jokaisen rivin päähän tämmöisen pienen höyhenen, joka puhalletaan kaverille ja katso kaveria silmiin. Yritetään saada se sinne jonon päähän asti. Ja sitten jos saadaan vielä takaisin, niin wow. You know, the little feather and you flow to the next one. Okay, we see that now the feathers are almost maybe run their course. Eli lähdetään toivottaa tervetulleeksi meidän päivän ensimmäistä puhujaa. Ja kuten kuulette, me puhutaan aika paljon englanniksi. Meillä on täällä lähes 30 ulkomaalaista vierasta. Meillä on vieraita Japanista, Norjasta, Alankomaista, Tanskasta, Tallinnasta, Latviasta, eli me huomioidaan nyt sitten näissä esityksissä myös nämä meidän ulkomaalaiset vieraamme. Ja Hasse Karlsson, professori Turun yliopistosta, aikuispsykoterapeutti,
aikuispsykiatrin ja tämmöisen niin maailmanlaajuisesti ihan todella kuuluisan Finbrain kohorttitutkimuksen johtaja on puhumassa meille hermostollisesta varhaiskehityksestä. So we want to welcome Hasse Karlsson, who is really a world-known scientist leading one of the biggest cohort researchers in the world called the Finbrain Cohort Research from Turku University. Hasse is, besides being a professor and a scientist, he's also an adult psychotherapist and also an adult psychiatrist. So, very warm welcome, Hasse, to our seminar.
Thanks so much for these kind words. An adult psychiatrist, but maybe not an adult. One more remark, I didn't realize that Jukka is that good in archery. I mean, that's really a skill. Congratulations. And congratulations also for the association for the anniversary. I'm really happy to have the possibility to address you here. I have also lots of time. I've never granted this much time for a talk. Was it 5 hours? But we can take a break at some stage, and you can ask questions and comment on whatever. So we can try to make this interactive, if possible. But let's start by warming up a little bit. Why the brain in the first place? Well, I mean, the brain kind of, it runs the show. It controls just about everything in the body. Even when you're asleep, you could actually maybe even say that you are how your brain works. And this is a trendy research topic nowadays, I think mainly because there are new methods to study the brain. We can image the brain in totally different way than for instance, for 20 years ago, so that's maybe one of the reasons. Then why study brain development? Well, of course, to understand the mechanisms of brain development and also the mechanisms of disorders, and to understand if something can be done. First of all, to prevent brain change, we know that there is brain change in relation to stress. For instance, I will talk quite a lot about that. And also maybe if there's possibilities to reverse these brain changes associated with adverse experience, for instance, and stress through different interventions, maybe even through psychological interventions. I mean, that's an area we don't know that much about currently, but maybe I have time to say a few words about that as well. Okay, so this is the little pig we are interested in, and it has been said that the brain is the most complex structure in the universe. I mean, think about it. There's probably around 80 billion neurons. Some say that maybe 100 billion neurons, and all the neurons have lots of connections with other neurons, maybe hundreds or thousands of different neurons. So it's a huge, huge system. You can just try to imagine how it looks like. The new thing is also that the brain is quite plastic. There's plasticity. That means that the brain reacts to environmental inputs and changes. It changes in structure and also changes in function. When I was a young MD, so in the med school, so I was taught, the teaching was going so that okay, we have the neurons we have when we are born. That we don't get new neurons if they are damaged, so you can't do anything about that and so on. But this concept has totally changed now in the direction of plasticity of the brain, and there's lots of development going on. For instance, during pregnancy, when there's a peak of neural development, so it has been estimated that 200,000 neurons are born every minute. One estimation is actually that about 50,000 synapses every second. So it's there's lots of things going on, and you can just imagine that how input into the brain also changes these systems then during development. Okay, so things have changed, and this was the situation until the 80s. The only thing we could do was to study brain anatomy, brain structures. Now the new thing is that we can also look into the living brain and see what's going on in the brain when people are thinking or having emotions or doing something, whatever. Although the things I will be talking about today is more or less like structural things, because in the developing brain there's not that much studies on these functional things. What are the methods? I mean, of course, the old thing is to use Röntgen devices, computed tomography, but the new thing is MRI imaging, which is actually the thing we are doing, for instance, in our cohort. The good thing is that you don't use radiation. You can do the imaging several times with with the same babies, for instance, because there's no long-term damage caused by the imaging. And then we have a long list of structural or functional brain imaging methods. Here's just at least I won't go into these technical details here, but I could just show you a picture of different devices. There's the MR device over there. This is a PET scanner. That's SPECT, that's transcranial magnetic stimulation, and so on. And then this thing, which has also been developed largely in Finland, MEG over here, and this is the zoomed in picture of the device. And you can clearly see here why MEG researchers call this the hairdryer from hell. Okay, this is what's going on in the brain during development. So one remarkable thing is actually the folding of the brain starts really late, in the late pregnancy, and then when we have a term baby, so the brain looks pretty much the same as in adult. Of course, the adult brain is bigger, but there's lots of structural development going on during pregnancy. Here's one of the challenges we have had in our cohort, for instance, because this is an MR image of different brains in different ages, and you can see that the colors are totally different here, and the contrasts are not similar as in the older brain, and this is because of the myelination. The myelination process is still going on in the child brain. But this causes really a big problem, because what is done with the brain scans in order to analyze the data, so you have to segment the different brain areas. You have to see where's the amygdala, where's the hippocampus, and so on, in order to, for instance, measure the volumes of these structures. There's softwares that do this, but they don't do it with kids' brains, because the contrasts are so different. So, for instance, what we have done with about 200 child brains is that they have been manually segmented, and that was about 2 years work for one person, full-time work to get that done. I will show you a picture of that a little bit later. So a huge amount of work. Okay, here's just a timeline of what's going on in the brain. Here's birth, and you can see that different thing happens at different times. Birth of neurons, apoptosis, the neurons that are not needed, so they will die. Synapses are formed, and here you can see the myelination starts a little bit before birth, but it's going on until adolescence and so on. So, and you can also kind of then think that these different processes are affected in different ways in different time periods, depending on what's going on in the brain during that specific time period. This is a kind of a idea of why, first of all, why we should intervene early, but also how the brain is more susceptible to changes early on. So the plasticity decreases during time and in increasing age. At the same time, you have to put more effort into changing the brain if you want to do that later on. And I'm sure that everybody has seen this James Heckman slide, which everybody shows everywhere nowadays. He was the Nobel laureate in economics, and his one of these ideas has been that the more you put impact into early interventions, so the more you get back in return as human capital, or even monetary things. Okay, so that's kind of the background, but what I will be talking about is this, and this is now, I try to show here that what we know about brain development in different phases. So this is like the postnatal brain, let's say the brain of a child, and these orange arrows show things that we know quite a lot about already. How early life stress affects the brain development, for instance, and how let's say, nurture or physical exposures and so on. But the blue ones are the ones we don't know that much about. And I will try to talk about early life stress and the effect on that on brain. Also prenatal stress, mainly how the mother is feeling during pregnancy. And then the really cool new thing is transgenerational epigenetics, because there's now studies showing that the exposures or the experiences of the parents even before they get babies, so they can affect the next generation in many, many ways. And that's something that's really interesting. And I try to go in this direction, because I will be talking about this first, because this is what we have been studying now, then a little bit about early life stress, then transgenerational epigenetics, and then if there's data showing that parent-child interaction can influence these processes somehow. Well, I will have to say that the data is very scarce. There's not much to say about that, but I mean, brain-wise, of course, there's lots of intervention studies otherwise, but not so much on brain, directly on brain development. So that's about how we will go through this talk. Okay, and if you think about brain and studying brain outcomes, so the clear thing, of course, is to look at brain structure and function. But I think these things later in the list, they are also like indirect measures of brain function. Temperament is very much biologically and genetically based. Cognitive development, child psychiatric illnesses, all are brain-related things. But I will mainly focus now on this thing, and I think that Riikka will talk about the rest then a little bit later. Okay, a couple of words of our study. We started to collect a big pregnancy cohort in 2010, so it's been ongoing now for nearly 10 years. We recruited pregnant women during the first trimester, during the first ultrasound investigation in Turku, around Turku areas. The basic idea was to try to understand the mechanisms related to child brain development, the topic I will be talking about today, and also then its relation to later illnesses. Obviously, I mean, psychiatric illnesses are there, but there's also studies showing that, for instance, early life stress increases the risk for cardiovascular diseases, even risk for Alzheimer's disease and so on. But we'll have to go on for quite a while to get there, because the kids are now 5 years old, so we have about 65 years to go. I will then be 122 years or something. So let's see. Okay, and the idea really is to follow up the kids for several decades, and we have currently 4,000 families in the study. That means 11,000 individuals, because we have recruited both the parents and the child to be born into the study. And currently more than 100 researchers and 44 PhD students in the project. So this is where we collected the cohort from. Turku is over there, Helsinki is somewhere east from Turku, probably over there somewhere, just outside the circle. We have also a couple of hundred families from the Swedish-speaking area, the Åland Islands, the islands between Finland and Sweden. So we have a small Swedish-speaking cohort there, from there as well. And if you're interested in what we are doing, so please check our webpage. We have also a baby gallery there, so we have informed the families who are in the cohort that if they want, so when the children are born, so they can send pictures of the child on our webpage, and there are a couple of hundred pictures of these nice. Yeah, I guess that this goes through right away into your hearts. Okay. And this is a multidisciplinary group. This is from one of our research seminars, something just to show that, I mean, this I think is one of the strengths, maybe also one of the weaknesses of the whole project, that there's lots of people from different areas. There's pediatricians, brain imaging people, cortisol, people measuring cortisol, and psychologists and so on. There's even one dentist. I don't know, I don't remember what she's doing. Oh, that's another picture of one of the seminars. Here you can see a timeline, what has been going on. So here's the pregnancy time. We have measured, for instance, how the parents are doing during pregnancy. 3 times this is questionnaire-based data, so these are gestational weeks over there, then the birth. And then there's lots of different brain imaging things that's been performed with the kids. I will show you a couple of pictures a little bit later. Lots of biological samples. There would have been too many arrows, so there's no arrows there, but there's blood samples and placenta and hair and lots of things. And then one of the strengths, which Reika probably will cover also, are the neuropsychological measurements of the kids, but also the parents actually, which is currently ongoing. And we are here now, so now the 5 years measurement point is ongoing, and it will be continue until next summer or something, and then we're done. Then we have a 1 year pause, then we start to collect data on 9-year-old babies. So to collect one of these time points takes 3 years, because it took 3 years to collect the baseline sample of the whole thing. So there's quite a lot of work to do with it. And there, just to show that, once again, what we have been measuring. Okay, but then, let's go into business. What do we know about these things? Oh, that was the idea why we are doing this. Well, and let's start with the prenatal period. And that's something, for instance, I have been really interested in for a long time, and it was a privilege to get the possibility to start studying these things as well. The basic idea is that many biological systems are programmed already during pregnancy. And this idea comes from studies from 1980s, epidemiological studies, where David Barker, for instance, showed that that if the child is born and has low birth weight, so that increases risk for many illnesses, like cardiovascular illnesses and so on. And this is a quote from one of his papers saying that common chronic illnesses, like cancer, cardiovascular illnesses and so on, they are not always results from bad genes and unhealthy adult lifestyle, but from poor intrauterine and early postnatal health. And this idea was that it's the the nutrients or the food that makes the difference. But we have been now interested in could also, like prenatal stress, have these kind of programming effects on the child brain, and that's what I will be talking about. This has then developed a movement which is called DOHA nowadays, Developmental Origins of Health and Disease. So there's lots of studies going on this. But the first question is, why does this programming happen in the first place? And I think this is quite interesting, because the idea is that it may have adaptive value because it attempts to match the individual's responses with the environment. But if the responses are mismatched, then disease risk increases. So you can clearly see this in the case of low birth weight, because the idea is here that if the fetus doesn't get enough food and doesn't develop normally, so it's a signal to the fetus that you will be born into a world where there's lack of food. And what happens then? So there happens lots of biological changes in the fetus, which kind of makes the system work very efficiently. And then the idea is that if the fetus is born into an environment where there is not food enough, so it increases the survival then in these cases. But if the child is born into a normal environment where there is food enough, so there's a mismatch, and then the growth is accelerated, and then there's health problems coming later on. Another question is that could prenatal stress carry similar signals, or of course not similar signals, but could it be so that if the mother is depressed or anxious during pregnancy, so that's a signal for the fetus that you will be born into a world which is a scary place. So you have to be alert, and all these stress systems, get programmed in the wrong direction in relation to the real world than where the child is born. So this is kind of the basic idea of this programming of also psychological things, and of course of the brain. And there's quite a lot of data showing that this programming really happens. This is one of the most or one of the best known examples, the Dutch hunger winter, which took place in the end of the Second World War, when the Germans blocked off some roads so that people in some areas of the Netherlands couldn't get food. 4.5 million people were affected, and about 20,000 people died. And now there's been researchers who have been studying the kids of the mothers who were pregnant during that time, and then followed up these kids now for over 7 decades. And there's lots and lots of findings. This is from a New York Times article last year, where they just, kind of put together what have been found. Lots of metabolic changes. Also, the kids of these mothers died younger. I think this is a better slide showing that there's huge amount of health risks or health outcomes among these kids. Coronary heart disease, diabetes, and so on, but also like brain-related things, schizophrenia, depression, lower cognitive function, and so on. So it seems that this environment where there's both high stress and low amounts of food lead to changes in the offspring. And how does this happen? This is a study where they measured DNA methylation of certain genes in the offspring of these mothers 6 decades later, and they showed actually that there's less methylation in certain genes. For instance, genes that program the development of many of the body organs, and that could be the reason why you can see so many different health outcomes or health problems among these kids. So it may be that the prenatal period changes the biology by changing these epigenetic processes, and then they can be long-lasting, even last for several decades later on. And of course, our question is that does this happen in the brain as well? I think I could maybe jump over. This is another project, Project Ice Storm, measuring the same things. And just to show you one thing, the difference between that one and the Dutch hunger winter was that in this project they could also disentangle the effects of objective stressors. For instance, days without power, they didn't have electricity for I think 1.5 months or something. And the subjective reactions, like the psychology of these things, by measuring, for instance, post-traumatic stress symptoms among mothers, and they also measured physiological reactions. And here the basic finding is just that you can here also see, even 19 years later, so you can see lots of changes in the kids, in their temperament, in their behavior, motor development, physical development, and so on. So it seems that also in these behavior-related, brain-related phenomena, so changes happen during these kind of exposures. But then how about milder things during pregnancy, like let's say mild, but let's say like anxiety and depression during pregnancy. So how does that affect the child brain? And there are some studies about that. This is one of our collaborators, Claudia Busch from Germany, where they showed, for instance, that if the mother has anxiety during pregnancy, mid-pregnancy, actually gestational week 19, and then you follow up the kids and compare the brains of the kids of these exposed mothers, so to say, and control, so you can see that in these red areas, the gray matter density is lower in the exposed kids. And actually this effect comes nearly totally from female brains. So you can see these red areas in the female brain are here, but they are lacking from the male brain. Of course, the problem with this study is that the imaging was done, 6 to 9 years after pregnancy. So of course you could say that there's something else, in between that has affected. And I will show you a little bit what we have found, because we have imaged the kids 1 month after pregnancy. So probably the postnatal period doesn't play any significant role there. They also showed that, the same group showed that when they looked at maternal depression, so they showed that maternal depression is related to lower cortical thickness in the kids in these blue areas, so the cortex is thinner in those kids who have been exposed. And they also linked this then with the child behavioral, child psychiatric outcomes. So I mean, if it's known for a long time, of course, that maternal depression is related with child behavioral symptoms. Here they measured externalizing behavioral symptoms, and that's known for a long time, but when they put into the same equation the cortical thinning, so this root was after that non-significant. So it means that maternal depression leads to cortical thinning in some kids, and that leads them to the symptoms. So we are quite near really understanding the mechanisms. Well, as near as we can get in human studies, because of course you cannot play around and do experiments with stress exposures in mothers and kids. And what is now how does this happen? One of the things that probably kind of drives these effects is cortisol. And we know, and probably all you know that I mean, if you have stress, if you have depression or anxiety, so usually the cortisol levels are higher. And cortisol is like an epigenetic machine, actually. It goes into the cells and it changes lots of the cell functions. And here they showed actually that when they measured cortisol levels in the mothers, this is at gestational week 15, and these are 4 different categories. You go higher in cortisol when you go in this right direction. And here also in the girls, amygdala volume was bigger the higher the cortisol levels in the mothers was, as you can see here. Quite linear, nice thing that you can see there. The funny thing is that here also in the boys it's totally messed up, as you know, the male brain is so. Okay, but here also the problem is that we don't know what happened after the birth during the 6 or 7 years period. But this is just a brief kind of explanation of the mechanism. So you know that when there's stress, so this axis, HPA axis, activates and the adrenal cortex starts to excrete cortisol. There's usually this negative feedback, but in certain situations, in depression for instance, it doesn't work, and then the production of cortisol is high all the time. And what does cortisol do during pregnancy? Here is an image of, this is the maternal blood vessel here, this is placenta, this gray thing, and this is the fetus. And these black things are like cortisol molecules. And now usually in this placenta, so you have this enzyme. We have, in the group we have tried to learn to spell the name of this enzyme, but it's totally impossible. So I call it nowadays 11 beta blah 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 enzyme. So that's the enzyme, but the enzyme breaks down the active cortisol into inactive cortisol. So I mean these white balls here are the inactive form of cortisol, and this is the idea is that it kind of protects the fetus from maternal cortisol. But if there's too much cortisol, so the limits of the enzyme is exceeded, and some cortisol enters the fetal side, and then can cause changes in the fetal brain, for instance. And of course, the other reason can be that the enzyme doesn't work well enough. Then also quite low amounts of cortisol can enter the fetus. And what is downregulation? This enzyme, maternal anxiety. There's clear studies showing that the more anxious the mother is, so the less activity there is in this enzyme. So it's kind of, there's a hit from 2 sides. The anxiety may increase the cortisol levels, and at the same time the anxiety decreases the activity of this enzyme. Also, if you eat lots of licorice, so that also downregulates the enzyme. And that's a new finding actually from people in Helsinki, showing that one of the compounds in licorice actually downregulates the enzyme. So that could be why things, or one of the mechanisms of why things happen in the fetal brain already during pregnancy in relation to maternal anxiety and depression. Well, what we have done now, I will show you a couple of pictures, is that we have done lots of brain imaging stuff with the kids. We have, for instance, used emotional stimuli in newborn babies, usually the day after birth, and measured EEG, event-related potentials on these babies. We have also performed MRI scans on 1-month-old babies. You can see there's a baby in there, this has been easy with the newborns because they are being milk anesthetized. The mother gives milk to the baby, and then the baby sleeps. And if there's a good ear protection, so they usually sleep throughout the imaging session. We have tried to do that with older babies, and it's totally impossible. I mean, we tried to do that with 1-year-olds and 6 months olds and 1.5 years old. It doesn't work. They wake up, you know. We try to do that during night time. It doesn't work. And now we are doing that with 5-year-olds, and that works well because you can, you know, they are compliant in many ways. And if you prepare the families and the kids well, so it goes well. We have been using optical imaging, which is where you use near infrared light. And shoot it through the skull, and it measures actually brain blood flow changes. This is a method developed in Aalto University here, and you can get, you know, these hemodynamic signals then. And here also we have been using emotional stimuli. And then there's lots of neuropsychological things going on, eye movement tracking. Riikka will cover these things. Lots of different kind of measurements. And now really we are measuring 5-year-old kids, and we have a really great research nurse, Susanna Silisala, who works with the families for, you know, days and days before the scanning. There's a mock scanner where they have all the sounds and they have the videos which they can watch there before they go into the real scanner. And now with the 5 years old, I think our success rate is over 90%, so it gets really... But it means that you have to work a lot, of course, to make them compliant. And of course, then if the kids want to interrupt, so we interrupt and so on. So that's... But just to show that it needs lots of work. So what have we found? Well, yeah, this is once again to show you the amount of work. So we have to segment the brain, you know, slice by slice in these 3 different planes, you know, by hand, and it's a huge amount of work. But then you get images like this where you can see where the thalamus is, where the amygdala is, where the hippocampus is, and so on, where the gray matter and white matter lines are going, and so on. And this has been done now, and we have been able to measure some things in the child brain. So the results now, which I will present, are from 1 month old kids. This is a study which will be now out in a couple of weeks, I would hope so, where we looked at amygdala volumes in relation to maternal stress. Actually, we measured depression symptoms and anxiety symptoms, put them together, and you can see that the more symptoms the mother has had during pregnancy, these are different trimesters, so the smaller is the male amygdala, the blue line here. Actually the female amygdala goes in a different direction, as is shown actually in the previous studies of this Berlin group, for instance. And the effects are there still after 4 years. These are 4-year-old kids, and here the kind of the exposure was just maternal anxiety. This is the line of the boys' amygdala, and this is the line of the girls' amygdala. So they go in different directions. I have to say already now that there's, of course, studies linking then amygdala volumes to different psychiatric illnesses, anxiety disorders, for instance, and so on. So this could be one of the early biomarkers of the risks of later illness. We don't know that in our cohort yet, but let's see. Then on this optical imaging side, this was I think this was 2 months old kids, where we also used emotional sounds, and the brain reacts differently to sad sounds in the kids whose mothers have had anxiety during pregnancy. In this area, this is a temporal parietal area, which has to do with auditory information processing, but also emotional information processing. So there seems to be functional differences there as well. Okay, and you can measure, of course, things on different levels. And this is from Saara Nolvi's thesis, actually. And we just show you this because of one thing. Maybe Riikka will talk a bit more about this, but there's also a finding that maternal prenatal stress is related to infant temperament, so that the more maternal symptoms the mother has, so the higher is the negative emotional reactivity of the child. So it seems that these prenatal exposures also kind of modify the child temperament into certain directions. These effects are not big, I have to say. Nobody starts to think that the fate of the kid would have been, established already during pregnancy. The effects are quite small, but you can think of them as like signs on the road, and these signs are showing this direction and these in this direction, and then after several years, if the development goes to the same direction, so then maybe we have problems. But I mean, this is not something that is like clinically significant at this stage. But why I'm showing this is that there's also something you can do about these things. This is not from our study, but I think this is quite interesting, because what they did is that they here also measured prenatal stress using the EPDS depression scale, and they showed that the more depression the mother has, so the more negative traits in the infant temperament you can find. So this is pretty much the same that we found. But then what they did is that they measured how much the mother is stroking the skin of the kids after birth. And the more the mother strokes the kids' skin after birth, so the less you have this effect. So this is actually, you could kind of say that the stroking buffers against the stress-related changes in the child temperament. And that's quite interesting. So it's also, if you think about possible interventions, so this is something maybe that can be done. And if you think about the mechanism, so what they showed here is that this is also a epigenetic change in the glucocorticoid receptors. I can't probably go into details here, but this is something that has been shown also in animal studies. For instance, if you look at rat mothers, so it's known that if the rat pups are stressed, for instance, they have been separated from their mothers, so they develop usually a change in the stress system. The HPA axis starts to work abnormally. But when they get back to their mothers, so if there's a mother rat who licks and grooms the pup a lot, so then these changes doesn't develop in the pup at all. And why is this? This is because it demethylates some of these glucocorticoid receptors in the pup's brain. So pretty much the same actually what has been found in the human studies on prenatal programming during famine, for instance. There's lots of animal studies on this, and really now the first human studies also showing that stroking the skin changes the epigenetics of the glucocorticoids, the stress system, and that's probably why you cannot see the same kind of changes in the kids then later on. We have been looking at stroking also, and this is an interesting thing, because what has been found from the skin, I think it's only about 20 years ago, so a new type of nerve cells which are called the CT afferents. So if you think about the different senses, so we have the normal afferents that tell you in what part of skin somebody is touching you and so on, but we have also these slow functioning nerve cells which are not myelinated at all. They're called C-tactile afferents, and they are activated when you stroke the skin. And you have these cells also only in the hairy parts of the body. So for instance, in your hand, what's this area in English? Palms. So you don't have those nerve cells at all. And you can think about that also from an evolutionary perspective. I mean, that's the hairy parts are usually the parts that the mothers of hairy animals are licking and grooming and so on. So you need them on those areas. So if you stroke the skin slowly, which means 3 to 10 centimeters per second, so then they activate. But if you do that faster, or if you do that slower, so they activate a little bit, but much less. And I think the main interesting part here is that what happens in the brain when you stroke the skin. We made an experiment in the MR device. One of our researchers, Jethro Tuulari, actually stroked the leg of the newborn baby with a soft brush slowly. And why was it the leg? Because you can't get further into the tube, so we had to do that on the leg. And the finding was interesting. I mean, usually when you touch the skin, so of course what activates is the somatosensory cortex, the yellow area here. And you know that in the somatosensory cortex you have this homunculus. So if you touch the hand of a subject, so this area activates. If you touch the lips, so this area activates and so on. But what activates after stroking? So it's the insula. And insula is a totally different area in the brain, and I talk lots about insula, but it's an area that has more or less to do with emotion regulation, for instance. It activates in relation to empathy, for instance. It has some cognitive functions, it has some stress reduction functions, and so on. So that could maybe be one of the mechanisms why at least some negative developmental things can be kind of at least postponed by stroking the skin. So this was from the fMRI study, and then we did the same thing with 2-month-old kids in the optical imaging thing. There we actually compared slow stroking and then fast stroking, and then again in the slow stroking group the insula activates. And this is really, easily reproducible, finding that can easily be reproduced. Okay. So that's about the prenatal things, what we know. There's of course lots of other imaging studies, but I think kind of the main areas have been covered now. It's the amygdala where the studies have shown most changes in the brain, some changes in the frontal cortex, some maybe in the hippocampus, but the amygdala seems to be the main area where changes are in relation to early prenatal stress. And then we can go over to the postnatal period, but I mean some questions or comments are welcome. Then I can have some water.
Yeah. Please feel free, all of you, ask questions. I have some questions. First of all, this is the brain-based evidence for parts of Theraplay, right? Everyone got that. Now,
I didn't say that.
Why do we use touch? Why do we use touch? I mean, because it has been the area that has been debated in terms of intervention research. So from our perspective, this is hugely important and hugely interesting. I would like to ask about the gender differences you mentioned a little bit. Sounded interesting. Like, could you say something more? It seemed like the boys are more susceptible to some kind.
Well, if you think about the amygdalas, actually the studies point into that direction, that the bigger amygdala probably is the vulnerable thing actually in the brain, and that, I mean, this is just speculation now, but why is it so that depression prevalence is twice higher in females than in males? Could it be that the female brain is more sensitive to effects of stress already during pregnancy? There's been some speculations about that maybe it's so that the male brain is more sensitive to other kinds of things, more harsh things, getting hit in your head or something, and these kind of more subtle psychological, psychiatric, symptom-based things could be the ones that affects the female brain more. That could be one of these, but this is just guessing at this stage. We'll see. In 15 years we know. So be patient. You will get the answers.
Yeah. I know this is a difficult question, but you talked about modulating factors, like when we think about prenatal anxiety and depression, it's really established. It's important to reduce that, because that will increase cortisol, and cortisol has really harmful effects for the fetus. Are there any modulating factors that would maybe lower the effects of the anxiety? I don't mean just intervention. I mean, we think about intervention. Do you know? You mentioned the Ice Storm study. They studied also personality factors and stuff like that. Do you know?
I'm not sure what do you mean by modulating factors? Rico will translate.
Thank you.
So yeah, perhaps like factors like maternal self-regulation capacity, or some like executive function. We have been interested in how these factors may modulate, and of course in postnatal in their environment. But I'm shortly presenting that perhaps the quality of interaction behavior after birth can and there is some evidence for that. And perhaps, but at least these two factors are what we have been interested in.
Yeah.
And especially I think these maternal, own regulated. Mom can be anxious, but still if she has a good regulation capacity or like executive functions and these kind of skills, they may help.
Okay.
Yeah.
That's helpful.
We are trying to
You know that better than I do. But during pregnancy, I have found a couple of intervention studies also showing that if you do something that decreases, for instance, anxiety during pregnancy, so also the cortisol levels go down.
Yeah.
And I mean and that's quite important because I mean it must be a biological thing that happens in the fetus that transforms things from the mother to the fetus. And it's not enough just to measure if the anxiety goes down. You have to go into the mechanisms as well. And this is, I think this was good news, anyways. In these, I think there's only 2 studies in the world about this currently. But hopefully more is coming. Okay, shall we? Yeah.
Can you repeat the ratio of speed to centimeters
Yes.
per centimeters?
3 to 10,
So can you demonstrate that?
Like this.
like this.
It's what we do in Theraplay.
Yeah.
Does it have to be on bare skin?
Sorry? On bare skin.
Does it have to be on bare skin or can you go through the shirt?
I don't know. I would guess bare skin is better, but I don't know. Yeah. You're experimenting on it.
Yes.
You feel better? Yeah.
I was very interested in the research about stroke as well, but I was wondering what if the child doesn't want the stroke, if you take neurodevelopmental disorder?
Yeah.
Is there any knowledge about
Okay,
that?
so the Leanne's question was, what if the child doesn't want to be stroked, but you mean like sensitive children who are like reacting so to stroke? Anything sensitive about it?
Yeah, autistic children maybe.
Maybe.
So what's the answer?
Sitting down.
I don't
So
know.
Leanne,
but,
is your question like, how should we do that anyway?
Well, I was interested if you have like 4,000 people in your cohort, maybe there will be some autistic children within that, and you have some.
That's true, but I mean this experiment, for instance, where we measure the brain responses, so that was done with 20, not with 4,000. Of course, we cannot do that with 4,000 kids. I don't know. I would guess that if you do that, you know, against the kids' will, so probably it's not a good thing. But I don't have any more evidence about that, anyways. Okay, then about the postnatal area. And if you think, where does the evidence come from? So there's lots of studies linking really traumatic experiences with brain development. I will show a couple of examples of that. And then there are studies of how the mother is feeling after the child has been born, so the postnatal mood, for instance, of the mother. And then there are just a couple of studies relating parent-child interaction with child brain development. But I could actually say that this is nearly non-existent. That was actually a surprise for me as well. And Rick, I think we'll have to do this.
Yes.
Yeah, but also look at the brain outcomes, I mean, really, and see what's going on. But if you start, but I mean, one example of postnatal interaction is, of course, breastfeeding. And there's one interesting finding from our study also, where we showed actually that the more cortisol there is in the breast milk, so the higher is the fear reactivity of the girls. So these are the fear reactive girls, and this is the cortisol level. Here again, nothing happens in the male. It's a funny thing. Why doesn't anything happen in the male brain? Okay, but the term here is lactocrine programming, and the idea is that maybe also the breast milk, you know, signals something to the child, and the child should change accordingly. And this has been also shown in apes. This is from I think what's this macaque apes or something. A relatively new paper here also, and the same thing they found here, that the more cortisol there is in the mother's milk, so this is something which they measured in the animals. They measured nervousness. I don't know what that is in practice in the animals, but probably the same thing, that the more nervous, the more anxious the small apes are, the more cortisol there is in the mother's milk. Here also the effect is much more clear in the daughters than in the sons. So this could be one example of some kind of programming also going on after birth in in the baby and then produced by the breast milk cortisol. Then if you go to these really traumatic things, there's lots of studies on the brains of kids who have been raised in orphanages in Romania or in Soviet and so on. I'll just show a couple of examples. There's studies showing that the prefrontal cortex is smaller among these kids. There's studies showing that the connectivity, this is uncinate fasciculus, this is the red thing that actually carries information from the limbic area, from the emotional areas in the brain, so to say, to the frontal cortex. So this doesn't work normally in kids who have been exposed to early deprivation, for instance. There's studies showing that the fibers doesn't, how to say this, the fibers are more diffuse, or the pathways are more diffuse in kids that have been exposed to social deprivation. So it seems that the brain is a bit more messed up in those kids as well. And there's Nin Tottenham studies, several studies, just show you one. This is kids who have been reared in an institution and then adopted from there. And the interesting finding was that if you look at amygdala, the red dot here, so only in the kids who have been long times in the institution, so their amygdalas are bigger. This is kids who have been there a shorter time, and then these white bars are like control kids from normal environments. You cannot see changes in the hippocampus or in the nucleus, in the caudate nucleus, but amygdala here again is bigger, and you can. It's known, for instance, that amygdala activates in negative emotions. I mean, if you have panic attacks, so your amygdala activates, and also in other negative emotions, you can see amygdala activations. So you can speculate that this is also some kind of plastic modification of the amygdala volume in relation to a bad environment, if you want to say so. And if you are there long enough, so then that develops. If you are adapted to a normal environment, so then that doesn't happen in the brain. And also one really interesting thing, usually when you think about adverse experiences, so it's something that happens to the kid, for instance. And this is a study where young adult men reported that they have been seeing violence. They have been seeing violence in between their parents, but nobody really touched the kids. So they have not been kind of directly exposed to anything physically. But seeing violence in the family several times leads to a change in this inferior longitudinal fasciculus. So this is the back of the brain, the visual cortex is here. So this white matter tract that brings or carries visual information from here to the limbic system, here again to the emotion centers in the brain. So that is abnormal. So just only seeing things repeatedly can change the structure of your brain. And the interesting thing here was that they showed that the sensitive period for these kind of changes is between 7 and 13 years. And you can kind of think that the kid probably is super sensitive to these kind of things in that age compared to a 3-year-old kid or something.
Hasse, can I ask, what does that mean in the behaviors of these children, if that part of the brain is impacted?
I don't know. Do you know?
I might guess, when we see children who are exposed to family violence, how they react differently, right?
I could guess that, I mean, seeing threatening situation would trigger really strong anxiety or something, especially in these kids. But I mean, this is a guess. I don't think they measured anything in the behavior part of the kids in this study. This is a meta-analysis of studies done so far on maltreated kids, and they try to put together all the findings, both structural and functional findings in the brains. And here are the areas that are affected, and the color codes go so that these pink areas are so-called frontal limbic areas. So there's the amygdala, hippocampus, there's the anterior cingulate cortex, which is the area in between the lobes, and then the orbitofrontal cortex. These are areas that are involved in emotion and motivation processing. So that, I think that's quite clear also from a clinical perspective. And then there's the blue areas, the frontostriatal areas. So there's basal ganglia, dorsolateral prefrontal cortex, the other part of the anterior cingulate cortex, the dorsal part, and then the cerebellum. And these are areas that are involved in executive functions, working memory, inhibition, and attention, also functions that you really need to function normally. So these two big areas are affected in relation to maltreatment in the long run. Okay, and this is actually the only study I found about interaction, because there was the term interaction in the title of my talk, but this is not that much I can say about this, because there are two studies out actually from the same group. Here they measured actually maternal sensitivity and then brain volumes in 3 to 6 months old babies. The number is quite small, and what they found was that lower maternal sensitivity is correlated with smaller subcortical grey matter volumes. And this is really, really difficult to understand, because I mean the subcortical nuclei really don't know what they are doing in relation to human behavior, for instance, and so on. So I don't know at this stage. They also measured paternal sensitivity. Actually, then I think the number was 29, and there was a finding that the cerebellum, the small brain in the back parts of your normal brain, it was smaller in those kids. And what that means, I mean, it's even more difficult to understand. But I guess that these kind of studies are on their way, and I think we would have actually good possibilities also in our cohort really to look at the brain structures in relation to parenting behavior, on the quality of interaction and so on, and especially on the effects of the predictability of the behavior. Rika will be talking about that a little bit later. That's a new really interesting measure of interaction, I think. Okay, and then the last part is this epigenetic part, but maybe if there's something somebody would like to ask at this stage, so please feel free. Yeah.
This situation of a differential between the male babies and the female babies, is there anything about the mother is female and the baby is female, so we have female to female bodies connected versus female to males?
Yeah, that's a great question. I don't know. We can't experiment on that, because I mean, usually males don't get babies, so it's.
Right, but I just.
That's a good question. I mean, that could be something which is hormonal. Yeah. That's I really don't know, but that's excellent question actually. It could be, and that could maybe explain the different sensitivity of the different genders, the fetuses. In relation to the exposure, for instance, but I don't know.
Well, Hasse, one thought that came up from your cerebellum with the sensitive fathers is that the cerebellar volume is, in animal studies, is clearly correlated with the amount of social play.
Okay.
So in a way, and we do know that paternal sensitivity is often expressed in sensitive play. So this is just an idea in the cloud that. Maybe that could be a reason why the cerebellum would want to have this playful interaction, and if the father is not sensitive, then it doesn't get the input it needs for development. Good guess. Yeah, could be. Yeah. Of course, if I remember right, I would guess that it's so that more play, bigger cerebellum. Yeah. And in this, it was vice versa. Vice versa. Yeah. So it's tricky to say, but I mean, this also kind of shows that, in a way, brain research is just taking its first steps now, and we are, looking at very, very, how to say it, like we cannot study brain in a so fine-grained way as we should do in order to really understand what is doing what in the brain and how it relates to behavior and so on. So we are, I mean, in 20 years people will laugh at us when we say that, they looked at amygdala volume, so that's total madness. I mean, amygdala has so many different parts and so on. But I mean, we have to start from something. Okay, and then the last short part, which I think this is the most coolest thing I think that's happened during the last years. So could it be so that the experiences of the mothers and fathers already before they get babies could be transferred to the next generation somehow? And here the concept is transgenerational epigenetic inheritance. I think I won't have the possibility to go into the mechanisms here, but the basic idea is that that if something happens during the pregnancy time, for instance, stress, nutrition and so on, so that can affect, of course, the mother, it can affect the fetus who is in the womb, and the fetus has its eggs already in the body, so it can also affect the third generation. And then these can be passed on to the next generations. In males, it's a bit more simple, as many things are, as you know, because here what is happening is that the male produces germ cells all the time, and germ cells are like open to effects of environment throughout the whole lifetime of the male. And the males doesn't have the kids there, so actually you just need 2 generations in order to show that these effects really happen. And I will give you a couple of examples on this. So, and this has been studied also in Berlin in our collaborators group, where they showed actually that they measured maternal exposure to childhood maltreatment. This was a questionnaire thing, so you can of course question that how well that measures things, but I mean they had a questionnaire measuring that how much maltreatment the mother had experienced in her own childhood. And then they linked that with the newborn brain anatomy. And they actually found, for instance, that the maternal who were exposed to maltreatment as child, so their child's brain were smaller. Actually, the intracranial volume was smaller. And this was actually because of there was less gray matter in the babies. You can see that they control for many different things. Maternal socioeconomic status, obstetric complications, obesity, recent interpersonal violence, pre- and postpartum symptoms, and so on. So, and this is so far the only study in humans showing this. But then the fathers, and this is getting interesting because it seems the fathers may have some role, you know, in family life. And this is really a new thing. We didn't know that before. Namely, there's animal studies showing that if you stress rats, male rats when they are kids, and then the male rats get babies, well, first of all, the HPA axis is blunted in the rats that has been stressed as kids, okay? And then these researchers took sperm cells from these rats and they actually identified microRNAs in the sperm. MicroRNA, like molecules in the sperm of the stressed rats. You didn't find these in the non-stressed rats. And what they did is then that they injected these just these RNAs in a zygote, which is like a fertilized egg. And then when this egg became a rat, so they had the same blunted HPA axis as the fathers had, and these were not related, you know, genetically to the fathers at all. So it seems that it was the sperm microRNAs that transferred this behavioral phenotype to the next generation, and that's cool. And we have found in our cohort, for instance, that we have measured paternal early stress with a questionnaire also, where you know measure physical violence, sexual abuse, and also emotional neglect and these kind of things. So we just calculated the number of these kind of events in the father's own childhood, and then linked that with the child brain, and these are these 1-month-old kids. And there was a clear finding, especially in the corpus callosum, that correlated with the paternal trauma scores, so to say. So here you have the more scores you have on this scale here, so the more you have experienced maltreatment, for instance, as a kid, and these are the FA values of the corpus callosum. So you measure actually the integrity of the white matter in the brain. And you can see quite nice correlation. Of course, here also it's not, the correlation is not huge, but it's clearly there. And then we found the same thing, and surprise, surprise, also in amygdala once again. So here are the early stress scores, and here's the amygdala volume, and you can see the bigger amygdala is, the bigger, the more his father as a child has experienced stress. And what's the mechanism of this then? So the thing that we have been studying, of course, is associative. We have data on the paternal childhood, but it's retrospective data, and then we have the child brain. We don't know how this goes. It can, of course, be that this is just genetics, that let's say fathers with big amygdala transfer this same phenotype to their kids. That could be possible. It could be also something which is called gene-environment correlation, which means that there's some studies showing that a child with a certain kind of genetic makeup has a higher risk for maltreatment. Let's say a kid who has ADHD, like Jade's, for instance. So it's quite clear that in certain parents, it probably increases the risk of maltreatment of that kid compared to other kind of kids. So it could be a gene-environment correlation, which actually then makes it understandable that why did a paternal stress course correlate with the child brain. But it could be also this germline epigenetic inheritance, so that the sperm line or the germ line of the father who have been maltreated has been changed. There's change in the sperm cells, and that carries over the effect to the next generation. The only way to know what is the mechanisms would be to show changes in the father's sperm RNAs. And guess what we are doing now? We are collecting these little guys from the fathers who reported high childhood stress themselves. And we actually, this is an interesting situation now, because in about 3 months we will get the results, and then we can see if it's so that really there are RNA changes in the sperm cells of the fathers who have been exposed to early stress, and then link this with child brain development, and let's see. Okay, I don't think I have much more to say. I could just conclude by saying that, well, it seems clear that stress in different stages of child development affects brain. That's quite clear. You can see both structural and functional changes in the child brain. The main areas or the main findings come from the limbic systems. I'm talking about amygdala and hippocampus. There's lots of studies showing changes there, and frontal cortex is another area, which is more like a regulatory area in the brain. And the interplay between these structures, of course, is quite important in the well-being of the child and later adult as well. These changes are related, at least to some extent, to childhood psychiatric symptoms. So there are some studies showing really that you can link the brain changes with the symptoms of the children as well. But we don't really know the long-term effects. So let's see if you can see that there's a bigger amygdala in kids who have been exposed to maternal stress, for instance. We can maybe see that this is related to some childhood behavioral problems, but what's happening in the long run, so we don't know at this stage. So we really need these long-term follow-up studies. And luckily, some of these are ongoing. And early interaction may also affect brain development, and the interaction may buffer against the effects of early stress exposure, but mainly this evidence comes from animal studies, and I think there's really need for doing these studies also in humans. There are, of course, studies showing that early interventions can change the behavior and do many things, but really looking directly at the brain, so that's non-existent currently. Okay, acknowledgements. These are the things, and then I will close at this time of year always with this picture. Thank you very much.
Okay, thank you so much, Hasse, for this presentation. I think my brain is like wired up. It's important, it's interesting, important to hear kind of like the latest science on brain research. Would it be okay if we would ask you to just share some thoughts with a colleague nearby, like what's in your mind, in your brains right now? Questions, thoughts, how all these beautiful facts, what did they stimulate in you? Do you have some questions? So let's do that for a couple of minutes, okay? Just share something. Some of the things that are in your mind and have some short discussion. Okay?
Okay. The tai me tässä mietittiin sitä, että kaikki mitä äsken kerrottiin, niin se on yhtä aikaa hirvittävän pelottavaa ja hirvittävän toivoa herättävää.
So what was said is frightening and exciting at the same time.
Not only exciting.
Yeah, but it's also, it makes us hopeful.
Okay, the comment I made was on Hasse's point that there was some correlation between cerebellar volume and paternal sensitivity, and I was just thinking aloud that in animal studies, cross-species studies, the size of the cerebellum correlates positively with the amount of social play. And then, of course, Hasse mentioned that here the correlation was opposite to what the animal things were. And of course, these are just, I mean, taking from cross-species studies into individual species development is not really the way to go. But it can bring up ideas, and what we were talking about was that sometimes it's not only the volume of something that is important, because there are many inhibitory functions also. So just the crude size doesn't tell whether it is functioning well or inhibiting well. And the sensitivity of the fathers might be able to regulate the cerebellum, and all of a sudden it might be that it doesn't need so many cells, like you showed the messy cell tangles of children from Romanian orphanages. They had a ton, and even with autistic children, they often tend to show extra volumes of myelin. So in a way, they haven't pruned enough. So maybe cerebellar pruning. So, but these are just thoughts.
Yeah, excellent, and really a good example of it that we really know pretty little. I mean, one thing that the males know is that size doesn't always make a difference.
Size doesn't always matter that much.
So
Jukka was commenting about
that relates to the brain as well.
Saitteko tekin Jukan kommentin suomeksi myös, eli se, että tässä on paljon tätä määrän ja amygdalan koon tutkimusta, mutta se, että se ei välttämättä tietenkään kerro, pikkuaivojen anteeksi, mutta se ei välttämättä tietenkään kerro nyt vielä kaikkia. Joo. Okay.
Mä kysyn suomeksi. Niin pohdin tässä sitä, että puhuit stressistä ja varhaisesta stressistä niin me tiedetään stressistä, että se on sopivassa määrissä hyvä asia ja sitten taas niin kuin liian vähäisessä määrässä niin kuin ei myöskään hyvä. Niin onko tätä nyt sitten tutkittu missä määrin? Että mikä olisi optimaalinen stressi?
Käännäksä vai?
Yeah, so the question was, we also know that too little stress can be not so good thing, harmful even maybe, I don't know. So what would be the optimal level of stress? Of course, how do we define stress in some of the cases too?
Yeah, that's a great question. Any other questions? But I mean, the important point is that you're right. I should have said that actually, because there are studies also showing, for instance, in animal populations, for instance, in monkeys, that they are reared in environments where there's no stress at all. So their systems are more or less like messed up. For instance, the stress system functions in the same way as in monkeys who have been reared in extremely stressful environments. So it's a U-shaped curve, and somewhere in the middle. So you have to train the stress system as well. But how much would it be? So and I don't know how to measure that even. So I mean that's a pretty tricky question, but really an important one actually.
And it's of course also depended on the individual factors. I mean, that's some are more sensitive to stress than others.
Yeah.
Could you say that they can't finish too?
Niin, että siinä sitten on tietenkin tosi iso merkitys niillä yksilöllisillä eroilla, että jotkut meistä kestää paremmin stressiä kuin toiset, ja sitten varmaan kliinisesti olisikin tärkeää osata bongata ne lapset, jotka on herkempiä.
Okay. Any other thoughts or questions you have in your mind? No. I think we were
discussing a little bit even when Hasse was talking that, and you can Hasse say if you don't agree, but we are making the leap from the research also to intervention. I mean, it really signifies we have to start early. I mean, the prenatal period, it's very important to start there. It seems, at least on the basis of your study group's research and others, in terms of getting the anxiety and depression down, it has such longitudinal consequences. I was really struck by thinking like it really affects the genes. It has these like transgenerational effects. So they are really important things to take seriously for us who work with parents and children. I mean, for politicians, you know, governing money, who will make decisions, which people we can help and in what kinds of ways. Yeah.
I think that's a good point. But there's also, I mean, what happens at least in Finland in the maternity clinics nowadays is that you screen for depression. Okay, and that's a good thing. But then in many places they don't know what to do next.
Exactly.
And they don't screen for anxiety, for instance. And actually, quite a lot of our results show that it may be that anxiety is actually even worse for the child development and so on. So I mean, there's lots of things really in practice to do also.
Exactly.
Sorry?
Could you repeat the last part you said?
The last part. About the anxiety. So some of our results also show that in terms of child brain development, so anxiety can be actually even worse than depression during pregnancy. And for instance, in Finland, depression is screened, anxiety is not screened, and you know the interventions are what they are. I've been talking with lots of nurses working in these maternity clinics, and in many places they don't say that. They say that okay, we can screen, but we can't intervene.
We can't do anything. I mean, I think many of us can relate to that, and you're right. It's not just depression. Often it's anxiety or a combination of these two things. So I think the word, early stress is important. So
there's actually a term which I think we should start using, which is toxic stress.
Toxic stress.
Stress is a bit, it's
that's
a bit
true.
blurry. Yes. But if you talk about toxic stress, so that's something which we shouldn't have, and the rest of the stress is not okay.
And that's understandable. Toxin stress. I mean, that's really what we should be looking for.
So I was asking for, and Hanna offered this to me. It's a great point to come in because the word toxic stress was coined by researchers in adverse childhood experiences, which you laudably are asking about, and you were referring to the amount of adverse childhood experiences, how they correlate to parenting in fathers and in mothers. And here I want to come back to the first commentator's point that this is really scary and really hopeful. And I think the hope comes much from the fact that any moment in time is appropriate for intervention. Because, I mean, you can always say this is too late, but it is not too late for the next generation. Working with seriously disturbed adolescents, you will be helping the next generation when they get children. And at the moment, my work is in violence services. So working with, as you mentioned, in the adverse experiences, violence in its various forms is the main that is one of the major aspects. And here. Working with children, with adolescents, with young mothers, with young fathers, with older fathers, so in a way, the hope comes from the point that into this system you can come at any moment, and you will have an effect.
Yeah, extremely important comment. Thanks for that.
Nyt kun on tullut tätä geenitutkimusta, niin ensin oli sitä tutkimusta, josta löydettiin niitä tiettyjä geenejä, joita ajateltiin periytyvän, mutta nyt sitten kun on tullut tätä epigeneettistä tutkimusta, niin onkin sitten tullut tavallaan näkyviin myös sitä, että se myöhemminkin vaikuttaa näihin geenien periytymisen prosesseihin, ikään kuin nämä geenien muuntumisprosessit ikään kuin periytyisi myös. Ja se tietysti Theraplayn tekijänä niin kuin kiinnostaa, että missä määrin semmoisia tutkimuksia on, että miten niihin ikään kuin prosesseihin voitaisiin vaikuttaa sitten myöhemmin. Kun se ei olekaan enää niin, että vaan ne geenit, mitkä syntymässä kai, vai?
Yeah, if I'm summarizing, tell me, Lena, if I'm saying wrong. The question is, how can we influence, the process when we see that the epigenetics show that the genes do change due to environmental pressure, such as toxic stress? So what can we do to reverse? How can we influence? Dave is answering for you.
Yesterday, I was talking with a colleague from another country at the Nurture and Play program, which Sarah and Hannah can tell you about. She was telling me about how there are many families without fathers present with the children. And there's a lot of research now that shows that fathers who are physically engaged with their children in respectful ways, have fun with their children physically, that the children become much better self-regulated. They do better even when they're working later on in life, and they do better cognitively in lots of ways. So how to find ways to help men who are in fathering roles play with their children constructively, and that would fit in with Yuka's observation and some of the other ones.
Yeah, but and going back to the epigenetic changes, so I don't think we know that much about that. We know that the epigenetic marks can change, but what do the interventions do? I have done one study for about 10 years ago where we showed, for instance, that depressed adults had lower amounts of serotonin receptors in their brains. That's known for a long time. Then we randomized them to two groups. The other one got psychotherapy 4 months, brief psychotherapy, the other one medication. And what happened after that was that the receptor amounts increased with 15% only in the psychotherapy group. And that must be an epigenetic change. Of course you could think of maybe some other mechanism. So it seems that it's also a plastic system, the gene, how the genome functions. So it's more plastic than we have been thinking about so far. But I don't think we know enough about what are the ways of intervening. Are there better ways of changing the epigenome than other ones? I don't know if you know about that. But I think, of course, we have clinical evidence, lots of clinical evidence that many interventions work. And if they work, so they have to change the brain, and if the brain changes, so it probably is an epigenetic change.
Right. Wow. Yeah. And just to say this once more in Finnish, because this is hugely important. This is, and also Hasse, your study is one of, worldwidely, it's rare. Eli Hasse on tehnyt myös sellaisen tutkimuksen, missä he todella vertas reseptoritasolla muutosta masennuslääkkeitä saavilla aikuisilla ja psykoterapiaan osallistuneiden aikuisryhmän välillä. He huomasivat, että eikö niin, että ryhmä, joka sai psykoterapiaa, niin heillä tapahtui näitä reseptorimuutoksia aivoissa, mutta ei sillä ryhmällä, joka sai lääkitystä. Se on itse asiassa ihan mielenkiintoinen, mikä se reseptorimuutos oli. Että käykää katsoa se artikkeli, koska silläkin on merkitystä, kun mietitään lääkityksen, terapian ja niiden kombinaatioiden suhdetta. Se on tosi hyvä. Tärkeä tutkimus, mutta kommentti on myös se, että me tiedetään kyllä, että interventioilla on vaikutusta, me nähdään käyttäytymistasolla muutoksia ja totta kai sen myötä me kehitetään koko ajan ja uskotaan meidän työhömme, mutta se, että on hirveän tärkeä ja mielenkiintoista saada tämä aivotutkimus nyt mukaan, että todella voitaisiin katsoa, että mitä siellä aivojen tasolla tapahtuu ja löydetään ehkä myöskin sitten uusia suuntaviivoja meidän interventiotyön tekemiseen.
Tämä on muuten yksi keino, millä tämän yksinkertaiset päätteet saadaan ymmärtämään asioita.
Joo.
Yksinkertaisia kuvia aivoista.
Yeah. So brain-based research, it's important. Okay, when we are developing interventions, and the comment was we have good interventions. We know that they work. We also have some empiria, even with Theraplay. We will talk about that later, and other kinds of interventions. We've developed something called Nurture and Play for prenatal period. We are trying our best, but what we would need to do in research, and hopefully in collaboration with Riikka and Hasse and groups like this, is also to show how do they work in the brain level? I mean, that's hugely interesting, not just for science. It will probably even help us to develop better interventions. So I thought that's why we need this collaboration and seminars like this. Right.
Hello. I'm Hanna Pinomaa, and I'm a Oh, I get nervous. Okay. I'm Hanna Pinaman. I'm a trainer in emotionally focused therapy. So I work with the couples. And the developer of EFT is called Susan Johnson. And she had done brain research on seeing how the brain can change with the couple after 20 sessions of EFT. So there is a brain research on the shift on that one. And as I'm thinking of how important it is to intervene, with the babies that we see, that the impact of what happens with the parents. So my dream and my goal is to intervene with the couples as young adults before they have families, to help them on their brain level to have that shift in their brain level, that they are able to build a secure connection with each other that has an impact on their brain, so they will have happier and babies into this world. Okay.
Joo. Tämä Hanna Pinomaa, EFT-kouluttaja, tunnekäskeisen pariterapian kouluttaja, niin tuo esille myös tämän parisuhden näkökulman, eikö niin, että vaikutetaan pareihin, heidän turvalliseen kiintymykseen jo ennen kuin syntyy lapsia. Ja totta kai ajatellaan tästä stressinäkökulmaa, niin voi ajatella, että sillä on ilman muuta niin valtava iso merkitys raskauden aikaiselle stressille, miten parisuhde voi, mikä tuki sulla sitten on. Mahdollisesti parempi tuki myös tästä parisuhteesta, että kyllä nämä asiat tukee toinen toisiaan jatkuvasti. Joo. Wow, interesting thoughts. Hasse, would you like to say something to closure your feelings? Well, I think we would like to say, Hanna, yeah. Thank you so much, Hasse, for coming. Thank you so much.